So students, how are you? Today in this video, we are going to discuss some important topics from the chapter Biomolecules of class 12 chemistry. This video will be divided into two parts. This is the first part that will comprise of the carbohydrates and basically the first part of proteins. So let us start and see what are the important topics of this chapter. The first topic that we are going to discuss here is the carbohydrates. Carbohydrates are obtained by the hydrolysis of aldehydes and ketones naturally. So they are also known as the polyhydroxyaldehydes or polyhydroxyketones. So directly or indirectly in our diet the carbohydrates are there. Not only us, the plants, the animals as well as us, all intake carbohydrates in the form of a food or somehow. Also some of the carbohydrates are obtained or initiated in our body itself. The carbohydrates are classified in three types, mainly monosaccharides, oligosaccharides and polysaccharides. This division is done mainly on the number of the carbohydrates that is present. As you can see from the first name, that is monosaccharide, it is clear that it is the simplest carbohydrate. It cannot be hydrolyzed or divided into simpler compounds. It is one only, that is one is the simplest carbohydrate. Examples are glucose, fructose, etc. The next one, that is the oligosaccharide. In this type of carbohydrate we can easily break it down into 2 to 10 monosaccharide. That is, it contains 2, 3, 4, 5, etc. up to 10 monosaccharides, and by hydrolysis we can break it. The examples are sucrose, maltose, etc. Also lactose as well, isn't it? The last one, that is the polysaccharide. So oligosaccharide limit is 10, means we can divide a oligosaccharide up to 10 monosaccharides. In polysaccharides we can divide it more than 10, means from 11 up to a large number we can divide it, creating more number of monosaccharides. The examples are cellulose and starch. Carbohydrates can further be divided into reducing and non-reducing sugars. All the carbohydrates, as it is given here also for your convenience, I have already written it, that all the carbohydrates which can reduce the Fehling solution and the tolerance reagent are the reducing sugars, and others that can reduce the Fehling solution and the tolerance reagent are the non-reducing sugars, isn't it? So this type of carbohydrates contain free aldehydic and the ketonic group. I am talking about the reducing sugar, that these reducing sugars or the type of sugar or the carbohydrates that contains a free aldehydic and ketonic group is a reducing sugar, and in case of non-reducing, they doesn't contain a free aldehydic or ketonic group. Mainly the free aldehyde or ketone depends on the disaccharides. In this case is not in the monosaccharide. This free kind of aldehyde or ketone is only present in the disaccharide and trisaccharide whatsoever, means etc. etc. Means but not in the monosaccharides. But in case of non-reducing, it doesn't present in anywhere. Downward you can see that here we have given some of the reactions, that is the preparation of glucose from sucrose as well as starch. You can see the reaction that sucrose when hydrolyzed give two kinds of monosaccharides, that is the glucose and the fructose. It should be noted that glucose and fructose contain the same molecular formula when they are in the condensed form, but when they are brought up in the angular form or structural form, the structures of glucose and fructose are little bit different, but in the formula, that is the condensed formula, is same for both glucose and fructose. Now talking about starch, when starch is being hydrolyzed, we get, as you can see here, n number of glucose. Basically, in our experimenting in a lab experiment, that is in laboratory, we do experiments in a small amount, right? So on that part from sucrose, we usually get two kinds of glucose, not two kinds, two glucose or two types of glucose, mainly alpha glucose and beta glucose. But in an industrial large scale process, we can get n number of glucose when we hydrolyze the starch. Understood? In this slide, you can see the structure of glucose. The left extreme structure, that is the open chain structure of the Fischer model. Here you have the D glucose and the L glucose. D stands for dextrorotatory, L stands for levorotatory. Or in common sense, you can say D stands for right, L stands for left. Basically, when in your hand you take a model of an electron or an atom, whatever, isn't it? An atom, a compound, so or something. Let us assume you don't talk about the chemistry. You take something. You take something in your hand, then to get a better view of everything inside or outside it, what you will do? You will turn it a little bit right. You will turn it left. You will turn upside down, isn't it? So we do the same kind of thing. Here we rotate the glucose on the right hand side, on the left hand side, and basically this is the first model or the structural model of glucose that we have achieved. On the right extreme, that is the ring structures, are the two different kinds of structures. Glucose is found to exist in this type of crystalline forms. These two structures, the ring structure, are the crystalline forms, which are also named as the alpha and the beta, isn't it? The alpha form of glucose, here the melting point is 419 Kelvin and is obtained by the crystallization of concentrated solution of glucose at 303 Kelvin. On the other hand, the beta form, that is the side one, the melting point is 423, that is it has little bit more melting point than the alpha form, and is obtained by crystallization from hot and saturated aqueous solution of at 371 Kelvin. You can see here that how we have achieved the ring structure without hindering the structure of glucose morely. It can be explained by the open chain structure of the glucose. You can see on the left hand side I also have given the open chain. It was proposed firstly that one of the OH group may be added to the CHO group and form a cyclic hemiacetal structure. It was found that glucose forms a 6-membered ring in which the OH bond will be at the 5th number of carbon, which is involved in the ring formation. Also, it can be explained that the absence of CHO group and also existence of glucose in two forms, as it is being shown here. Just see the diagram, you can understand it. Till now in this video, we are discussing how we can break a disaccharide, how we can break a trisaccharide. So before breaking it down, except the monosaccharides, the disaccharides, the trisaccharides, tetrasaccharides, pentasaccharides, etc., up to the end of oligosaccharide, or also the polysaccharide as well. So all these plural number of saccharides are what? Here only the monosaccharides are being bonded, isn't it? Every di, tri, tetra or a polysaccharide is the plural form of monosaccharide. Means we are joining the monosaccharides. So how these monosaccharides are being bounded? Two monosaccharides are joined together by an oxide linkage formed by the loss of a water molecule. So in order to achieve something, we have to eliminate something, isn't it? So this kind of linkage is achieved by the loss of a water molecule. Such a linkage between the two monosaccharide units through the oxygen atom is called the glycosidic linkage. See downward. Here I have given a figure of sucrose. Sucrose means what? When you hydrolyze a sucrose, you get a glucose and a fructose, isn't it? So here, as a whole, the sucrose, on the left hand side you have the alpha D glucose, on the right hand side you have the beta D fructose, and they are being binded by a CHO structure. The two monosaccharides, namely the glucose and the fructose, are held by a glycosidic linkage between the first carbon of the glucose and second carbon of the fructose. That is, the link between the two, that is the glucose and the fructose, is being achieved when the first carbon is being linked to the second carbon of fructose. Since the reducing groups of glucose and fructose are involved in glycosidic bond formation, sucrose is an non-reducing sugar. Similarly, after sucrose, here I have given some more important structures together with their linkages. For maltose, the glycosidic linkage is present between the first carbon of the glucose with the fourth carbon of the another glucose, because when you hydrolyze maltose you get two types of glucose, isn't it? So here carbon 1 linked with the carbon 4 of the another glucose. Downward, in lactose you have one galactose and glucose. The glycosidic linkage is present between the first carbon of the galactose together with the fourth carbon of glucose. Now let us come to the polysaccharides. We all know the polysaccharide contains a large number of monosaccharides binded together by a glycosidic linkage. The first and the common polysaccharide is what? Starch, isn't it? So starch is mainly present in the plants. They are the main storage polysaccharide of the plants. Also, they are the most important dietary source of our human beings. The starch is made up of mainly two kinds of things: amylose and amylopectin. Let us assume in our body we have a large intestine and we have a small intestine, isn't it? So large intestine occupies more space and the small intestine occupies less space. Similarly, it is the case of the amylose and the amylopectin. The amylose is water-soluble component which constitute only about 15 to 20% of the starch. Chemically, if we want to say, amylose is a long unbranched chain. It contains 200 to 1000 alpha-D glucose units, means C. Here in polysaccharide, we don't talk about 10, 20, 30. No, we are talking about 200, 1000, 10,000. This kind of monosaccharides bind together to form a polysaccharide. So only in 15 to 20% starch we have 1000 alpha-D glucose units, and they are also binded by glycosidic linkage between the carbon 1 of the first monosaccharide with the carbon 4 of the second monosaccharide, and so on. Carbon 1, carbon 4, carbon 1, carbon 4, so on. Now talking about the amylopectin, that is the second one. Amylopectin is totally different from the amylose. They are insoluble in water, and it constitutes about 80 to 85% of the starch. Means a major amount of starch is being present in the amylopectin. It is a branch chain polymer of alpha-D glucose units. Here also the glycosidic linkage is present between the carbon 1 and carbon 4 of the alternate monosaccharide. One more important thing about it is that in amylopectin we have branch. With an open chain or a straight chain, we also have the branch. These branches were formed of the carbon 1 and carbon 6. Means on the main chain they will be on the carbon 1 and on the carbon 6 of the branch 1. Either it goes vertically or horizontally. The next important polysaccharide is the cellulose. It is exclusively found in the plants only. It is a straight chain polysaccharide composed of beta-D-glucose unit, which are joined by glycosidic linkage between the first carbon of one glucose unit and the fourth carbon of the next glucose unit. Coming to the next important topic after carbohydrates, we have the proteins. Like carbohydrates, it is also an essential part of our human life, also of the plants and animals. The proteins are the most abundant biomolecules of the living system. We can achieve this kind of proteins that our body requires from many food that we intake, like milk, cheese, pulses, peanuts, etc. They occur in every part of our body and form the fundamental basis of structure and functions of life. The first subcategory of the proteins are the amino acids. Amino acids contains an amino group and also a carboxylic functional group. These subcategory amino acids are also essential part of our human life. There are two types of amino acids: essential amino acids and non-essential amino acids. Here in this clip already I have distributed or categorized the essential and the non-essential for your convenience, such that you can differentiate it easily. So what are these essential amino acids? The amino acids which cannot be synthesized in the body and must be obtained through a diet are the essential amino acids. On the other hand, any type of amino acids or the important amino acids which are synthesized in the body are known as the non-essential amino acids. We have a list of amino acids that is present in your book also. Out of those, some are essential and some are non-essential. In your book, you can find the chart that contains all the 20 amino acids. Out of that, we have 9 essential amino acids. What are those? Those are the histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan and valine. Except these 9, all are non-essential amino acids. For your convenience, in this video also I have just noted down the names, the structures and their abbreviations. All the 20 are here. The green part, you can see here, the green part are the essential amino acids and all the black ones are the non-essential amino acids. Coming to the next topic, just see on the left hand side of the clip. Here you can see there is a structure and there is a definition. The structure is of amino acid and the intermediate ionic form of the amino acid, also known as the deuterion. It may be noted that for 1 mark or somehow the deuterion comes. Okay, till now all the things that we have discussed, the structures of the maltose, lactose, whatever, also the difference between the reducing and the non-reducing sugar. Now the deuterion, these are important questions for your boards. So amino acids are usually what? They are colorless and crystalline solids. These are water soluble, also high melting solids that behave like salts rather than simple amines. This kind of behavior is given because, as I have already told you, it contains a carboxylic group and an amino group. See here, the carboxylic group is a acidic substance and the amino group is a basic one, isn't it? Means that amino group has one acidic part and one basic part. In aqueous solution of the carboxylic group can lose a proton and the amino group can accept a proton, giving rise to a dipolar ion known as the deuterion. As it is an acidic group and a basic group, that is, it has a positive and a negative charge, but it will be a neutral one, although it contains both the positive and the negative charges. The positive one will dominate the negative one will also dominate the positive one, making it a neutral. In spite, it contains both. In the deuteric ionic form, you can see on the right hand side the deuteric ionic form, the amino acid shows the amphoteric behavior. And you know the definition of amphoteric also, isn't it? That is, it shows both the property of acidic and basic. After that, here, depending upon the structures of proteins, we are going to say something. Like in carbohydrates, you have the glycosidic linkage. In proteins, you have the peptide bond or the peptide linkage. So, on the basis of the molecular shape, we can divide the proteins into two parts, that is the fibrous proteins and the globular proteins. So, what are these fibrous proteins? The polypeptide chains that runs parallel and are held together by hydrogen and disulfide bonds. Remember, there are two kinds of bonds, either the hydrogen and the disulfide bonds. Then the fiber-like structure is formed. Such proteins are generally insoluble in water. Examples are what? Keratin, that is the most common example. Your hairs, isn't it? It is found in the hair. Keratin is found in the hair. And myosin, that is present in our muscles. Talking about the globular proteins, obviously it is different. It's like a spiral one. The structure results when the chain of polypeptides cell around to give a spherical shape. They are usually soluble in water. Not unlike that fibrous protein, they are soluble in water. One of the commonest example of globular protein is the insulin, and the albumins as well are the common examples of globular proteins. The structure and shape of this kind of protein, that is the globular protein, can be studied in four different types or four different levels: primary, secondary, tertiary and quaternary. Here you can see the diagrammatic representation of all the four structures that is given here, which are colored balls represented by an amino acids. The primary one is a simple sequence of a chain of amino acids, while in the secondary one, the hydrogen bonding of the peptide backbone causes the amino acids to be folding into a repeating pattern. Talking about the tertiary one, the 3D folding pattern of a protein due to side chain structure interactions. Now lastly, the fourth one, that is the most puzzle one, right? The protein consists of more than one amino acid chain. It's not only one. See, you can see there are two kinds of colors right here. So we have more than one kind of amino acid chains present in the quaternary protein structure. So thank you guys. So in this video you have learned a little bit about the carbohydrates and the proteins. In the second part you are going to learn the rest of the part of this biomolecules chapter, that is of the amino acids and other essential topics. So what are the topics? That is the enzymes, vitamins, etc. So till then, ta-da.